Now, as I alluded to in the last lecture, it's not only the excited electron that's going to be a short-term energy source, but it's the making of ATP, which is what respiration is driving towards. ATP, as we learned in the last chapter, in chapter 6, it's -7 point whatever kilocalories of blah, blah, blah that it brings and lowers the delta G. So, how do we get it? What we find, as you see here, is that third phosphate of ATP, adding it on there, and doing so in such a fashion that the second law of thermodynamics is lowered. Okay, you can't get rid of it, but you lower it to the point where adding that third phosphate, you're only going to lose a certain amount of energy to do so. As you add it, energy will be stored, the majority of it will be stored, so that you can remove that third phosphate at a later time point and transfer that energy to something else. As you see, as I pointed out, this reaction. The green ribbon's a polypeptide. It's part of an enzyme. Enzyme needs to be able to do something. So what do? You pop off that third phosphate. You add it to that polypeptide. You've phosphorylated it. By phosphorylating, you're taking that delta G of -7.4 or 7 point whatever, and you're now adding it to that polypeptide that's part of a much larger enzyme. So you're now adding it into the mix. That's how ATP works. ATP is not this magical battery. It's not this magical thing. It's just taking a high energy phosphate and transferring it from the mid-level storage device of ATP to an enzyme. So this phosphorylation, transferring the phosphate from ATP to its target, whatever the protein, the enzyme, the glycoprotein, glycolipid, whatever it may be, we call this substrate level phosphorylation. Substrate level infers that this is an enzyme, because remember, you see enzymes work on substrate, take substrate, turn it into product. Here, the enzyme is recognizing some substrate, and it's taking ATP, popping off the phosphate, and adding it to it, phosphorylating it, phosphorylation. As we go through and we look and we see the making of ATP in this chapter, the breaking down of macromolecules that you've ingested to transform that energy into ATP, we're going to see it's going to happen in one of two ways. First, substrate level phosphorylation. Okay, gonna take substrate, pop off phosphate, add it to ATP, a diphosphate. It's very slow, very clunky. Second method is what we're going to see later in this chapter is known as chemiosmosis. Chemiosmosis is basically a process that is very, very fast. Substrate level, slow and clunky. Chemiosmosis, very fast, very thorough, little to no waste. A lot of what we're going to be talking about in this chapter is going to be occurring within the mitochondria. The majority of the ATP that's produced in your cells occurs in the mitochondria. That's why they've given it the nickname, powerhouse of the cell. It makes the ATP, the majority of it, not all of it. We're going to see that ATP can also be made to an extent in chloroplasts and along the plasma membrane of prokaryotes. The levels in chloroplasts, the levels on the prokaryotes is nowhere near what you'll find in the mitochondria.